So everybody, if I could just make a little bridge to from Rabbi Harkzak's topic to my topic. My daughter's at the other end of women going into labor. She's an OBGYN, and sometimes she has to drive to the hospital on Shabbat to deliver babies. So that probably raises a whole bunch of other halachic questions. But to tie it all together, a number of the mothers that she has to deliver right now are like also COVID positive. So it's sort of like a little bit of a bridge between the halachic questions and my topic, which is on the coronavirus. So let me just say a couple of things, as we get started. One, I think that among everyone listening, there are people who have like better or not as good backgrounds in science. So I just want to say that if anyone has any questions as I am going along, don't put them into chat because I don't try to read chat during presentations, but just unmute yourself and ask the question. If we start using up a lot of time with that, I might ask everyone to just hold the questions until the end for a Q&A. But as of now, any questions come up, please just unmute yourself and ask the questions. Second thing that I want to point out is that some of the information I'll be presenting is based on information which is coming out of recent journal articles that are not peer-reviewed. This is something which is really new to to a lot of researchers. Anytime a researcher wanted an article published, they would send it into whatever journal they were interested in having the article published. And there'd be a whole review process where the article would be sent to various research scientists in the field. And if there were any issues, it might have to go back to the author for certain revisions. Now, a lot of the articles that have to do with COVID-19 are being released pre-review, and the articles are not, the reason they're doing that is they want scientists to have access to all the information possible with the understanding that the articles have not been reviewed. So this is, so some of the information I'll be presenting now is based on that type of situation. So let's get started. So it's interesting to note that this coronavirus, SARS coronavirus 2, is a tiny, 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 tiny organism. It's hard to imagine how small it is. Under the most powerful regular microscopes, you would not be able to see one. You really need an electron microscope. The diameter, these are spherically shaped viruses. Their diameter is about 120 nanometers, and a nanometer is 10 to the minus 9th meters. So we're talking about viruses which are unbelievably tiny. And whenever I think about this, I kind of feel that it's a little bit ironic that we have weapons now that can kill hundreds of people within seconds. We have bombs that could destroy battleships in seconds. We could blow up whole cities in seconds. These little teeny, 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 teeny viruses, we're struggling for a way to defeat them. And so what I'd like to sort of get across as we go through this is, how these viruses work and why it's so challenging to deal with these particular viruses. So some of what I'm going to say, might be stuff that everyone knows, but again, I think everyone has like different science backgrounds. So try to, be, patient. So let's get started. And so I'm going to need to change screens a few times. Right now you're looking at a PowerPoint, but I'm going to need to show you some other, I'm going to need to go out of the PowerPoint and use some other programs during the presentation. Okay, so All right, so just some very basic terms, and you might be very familiar with these, but I'll be using them during the presentation. A pathogen is any disease-causing organism. So there are many viruses which are pathogens. There are many types of bacteria which are pathogens. There are other organisms that could be pathogenic, like mold, various types of mold, and there's many, all types of fungi. There's many different types of disease-causing organisms. Antigens are molecules, generally on the surface of these pathogens, that initiate some type of immune response. So if you look at these little spherical structures, these are the coronaviruses that we're dealing with now with this disease. The antigens are these little green structures, which I'm going to show you in more detail shortly, but these little green spikes, which is where the corona, the term corona comes from. Okay, so the pathogen is the organism, the virus. The antigen are these little green proteins that they're showing on the surface. And then there's another term called epitope. One antigen could be a fairly large chemical. We'll see that these little spikes that are on the surface of the coronavirus are made up of tens of thousands of atoms. So they're from a molecular standpoint, they're pretty large. And there could be different regions of the antigen that elicit an immune response. And those regions will be referred to as epitopes. So as I wrote here, a pathogen could have many antigens, and antigen could have multiple epitopes. And this is really important to people, to researchers now who are working on finding a way of making antibodies, of getting us to be able to make antibodies against these various antigens. So just a little bit of a background in terms of our immune system. It would take days, if not weeks, to really do a decent job of explaining how the immune system works. But I just wanted to show you a few quick things. This is a diagram that I give to my AP Bio students, which summarizes what we sometimes, it is like referred to as the adaptive immune system. We have innate immunity and we have adaptive immunity. So this is really more about the adaptive immunity. When our bodies are exposed to antigens, which I'm showing up here, and you sort of like need to follow my little arrows as I take you through diagrams. But when you're first exposed to an antigen, that causes what are called B cells in your body to start multiplying. And they multiply into two populations of cells. One population becomes what are called plasma cells. And those are the cells that secrete these Y-shaped antibodies, which protect you against disease. The B cells also divide into what are called memory cells, memory B cells. And so if you should ever become infected with this pathogen again, which could happen, you'll have so many what are called memory cells that could be converted into plasma cells so that you would fight the disease without even being aware that you were infected again. Some people, I've heard some people say, oh, you can't get the same cold twice. You could get infected by the same, let's say, cold virus more than once, but the second and third and fourth time, you'll have so many memory cells that you'll be able to fight it off quickly, so quickly that you wouldn't even know that you were infected a second time. You wouldn't even get to feel the symptoms. So the B cells, again, work against, they recognize antigens, and in a number of different ways, they'll work to sort of neutralize the effect of those, the pathogens that those antigens are attached to. So in terms of viruses, the B cells make antibodies which attack the viruses directly. On the other side, all right, our cell-mediated adaptive immunity involves what are called cytotoxic T cells. And these cells, these immune system cells, which mature in the thymus, that's why they're called T cells. So these cytotoxic T cells will destroy virally infected cells. So if you have a cell that's infected with a virus, that cell is not going to survive. That cell is eventually going to burst open and release a lot of viruses. Viruses multiply within cells. And so once the virus has successfully entered into the cell, whoops, got to go back. Once the virus has successfully entered into a cell, that cell is doomed. And so even though it may sound terrible that you're destroying one of your own cells, you'd rather destroy that cell while the viruses are still forming rather than allow that cell to burst open and release many viruses that will then go on to attack many other cells. So we really have like this two-pronged attack for destroying viruses. Antibodies, which will destroy the virus, or cytotoxic T cells that will destroy virally infected cells. And I didn't point this out, but you also make memory cytotoxic T cells. So that again, if you ever get infected a second time by the same pathogen, you will respond to it so quickly that you won't feel the symptoms from that attack. Okay, so that's just a very quick summary of some of the major things you need to keep in mind regarding the immune system. There are also things called helper T cells, which stimulate both sides of the immune system. Okay, any questions so far? If anyone, again, if you have any questions come up while I'm presenting, don't hesitate to unmute yourself. Okay, so let's go on to the.
I have a question.
Yeah.
Hey, Bob. John Kroll.
I have no idea who you are, but.
It's John Kroll. You know, we work together.
Oh, John. Yeah. Thank you.
Yeah. I noticed that, you know, in hospitals, they're taking plasma cells from people who have been infected and using them as treatments.
Wait, plasma or plasma cells?
Oh, plasma, I believe.
Yeah, because that would be like the liquid portion of your blood that might have antibodies in it.
So would that be, that's regarding the left side of the screen. Are we doing any sorts of, you know, treatments regarding like the cytotoxic T cells, or is that not something that's done in terms of therapies? Usually
we work from the standpoint of making the antibodies. Like when you get immunized, you're given like a harmless form of the pathogen. And then what your body does is it makes it will make plasma cells and memory cells to destroy it. But the good thing when you get immunized is that you have these memory cells, which for many diseases will give you lifelong protection. So that would be that's like the main thing that they're doing when they're giving you what's called active immunity. Now, sometimes you could be given what's called passive immunity, where you're actually given the antibodies. And that type of thing would be done if it was a disease that, there are certain diseases, you have to think of it as like a race between the pathogen and your body. And in most cases, your body will destroy the pathogen before the pathogen destroys you. But for some diseases that are very, very, where the onset and the disease is very serious, there are certain cases where there's a real danger that the pathogen will kill you first. And so there are certain situations where they'll actually give you injections of the antibody, which is giving you passive immunity. It doesn't doesn't cause you to make memory cells, so it doesn't give you lifelong protection. It only lasts as long as the antibodies last in your body, which is not that long. But that helps you fight the pathogen, which might otherwise kill you. So there are certain situations where they take that approach. Okay.
Hey, Bob, I got a question.
Yeah.
Is there a concept of immunity that doesn't involve antibodies? Meaning, can you be immune to get it the first time?
Could you be immune?
Meaning, are there people who don't catch colds, or are there only people that have antibodies that kill the cold virus?
No, I mean, everyone, you're not, well, there are some antibodies that are able to cross the placenta. So you might be born with antibodies, but generally speaking, the antibodies are not that, they don't remain in existence that long. But the memory cells do. Just to give you sort of like a timeline, if you could sort of picture this, let's say you were infected. So it might be like 4 or 5 days before you feel the effect of that infection. So you're feeling fine. You don't know it, but you've been infected. Somewhere like within those first 5 days, your body recognizes the fact that you're infected, and you start making the B cells. And if it's a viral infection, the T cells will start multiplying. And so there's a certain point where there's sort of a battle going on between the beginning of your immunity, getting higher and higher. And I don't know if you guys could see me, but the immune system becoming more prepared. But at first, the pathogen is sort of winning out. And you might be sick for 3, 4, 5 days, maybe a week or so, whatever, depending on the pathogen. But gradually, you build up a lot of antibody. And so you begin to win out, and you begin to gradually feel better. So depending on the pathogen, the time frame for everything I'm saying could be a little different. But you begin to win out over the pathogen because your immune system is fighting it off. Interestingly, when let's say you get infected with that same pathogen a month later or a year later or whatever, you now have all these memory cells. So in this case, if you get infected with that same pathogen, those memory cells, instead of taking, let's say, about a week from the time they begin to make antibody, and it might be like 10 days after you were infected, instead of it taking that long to make a certain level of antibody, in about 2 or 3 days, you'll make that amount of antibody. So let's say it would normally take 4 or 5 days before you began to feel the infection. You're destroying the pathogen before you even feel the symptoms. And the level of antibody might be 2 or 3 times higher than it was the first time you were infected. So that's why you could be infected with the same pathogen multiple times, but you're not going to get sick from it multiple times in most cases, all right? We're hoping that that's true for any immunization that's made against this, nasty coronavirus. If you look at this chart, this is a chart that I gave out to my AP Bio students when we were discussing viruses very early in the year. I think probably around October, maybe the beginning of November. And, I just showed them like different types of viruses. Some viruses have DNA in them, some have RNA in them. There are different types of DNA and RNA that you could have. So I was just showing them, I gave them a few examples. I pointed out a few of the viruses and certain types of illnesses that they might have been aware of. And I sort of, I chuckle a little bit looking back at this, but this one, I just passed through, barely mentioned this coronavirus. I had no idea back in October that if I were teaching this today, we would do a full stop at this virus and spend, a lesson or two just discussing like how it works. But it was just another virus on the chart back in October. All right. So let's go on. All right. So this is the virus we're talking about. All right. This is a diagram of the virus that causes COVID-19. A few things I want to, really point out about it. One is the surface, and this we're going to spend a good deal of time looking at, but on the surface are these so-called spike proteins, which is really what the term coronavirus is based on, corona. These spike proteins are huge proteins. I don't know how much you remember from your days in school, but the proteins are made of building blocks called amino acids. This spike protein, as it's called, Sometimes in literature, they just put an S to refer to this protein as opposed to other proteins that are in this virus, is made of 3 chains of amino acids, several thousand amino acids, and tens of thousands of atoms. It's a huge molecule. And we're going to see exactly, in a short while, we're going to see what this spike protein does. I do want to point out that inside this type of virus has a lipid membrane. Not all viruses do. Some just have like a protein coat. But this has a lipid membrane. It has this region called the nucleocapsid. And in that region, in here, is the genetic material of the virus. Now, I'm sure you remember from high school or whenever that the genetic material is DNA in most organisms, but in some viruses, the genetic material is RNA. And in this particular case, the genetic material is RNA, what we call the positive strand of RNA. This is RNA that actually codes for some of the proteins you see either on or in this virus, all right? What I'm going to do, well, I'd like you to see, but let me just, well, I'm gonna go, let me go back a little bit. I'm going to take you into another program, so it'll take me a second to move into the other program, but I want to show you these viruses, the spike protein, in a little bit more detail. So I'm going to move to, this is a program. Is this taking up like most of your window?
Bob, it's great.
It's great, you can see it? Okay.
Yep.
So this is the spike protein. You're looking like down into it. And this is a program which I now teach the AP Bio students to use for doing all types of modeling of different types of proteins. And you can really like manipulate the protein. There's all types of little things you can do. This is the basic position that it would be in if you were looking at that spike protein that I showed you in the last slide. If we're facing vertically, this is how it would look. It's a little wider in this area and a little narrower down here. And it's made of 3 chains of amino acids. And I'll just show you those. I'll just sort of highlight those 3 chains for you. This is the A chain. Oops. Okay, this is the A chain, all right? And then there are 2 more chains, which I'll show you in a moment. That's another chain. And then the other chain is all the parts that I did not color. There's a lot of different things that you can do, with this program. I'll just color the other chain just so you can see it clearly. They're just called chains A, B, and C. So I'm just typing in the instructions. Okay, so now you can really see the 3 chains. You may notice it's a little difficult to tell. Let me shrink this down just a little bit. But this part of the A chain is sticking up a little bit more. Try to remember that, because when I show you how this virus infects cells, 2 of these chains are going to project a part of their structure a little bit further out to hook up to the cells that they infect. So I just wanted to sort of highlight that. And now I'm going to, we may come back to this program again, but I'm going to go back to the PowerPoint now. Okay, so that was this structure that we were just looking at. Okay, so now we're going to move on. These are screenshots I took of that protein. There's something else I wanted to point out to you. So this is just the first chain that I highlighted, and I was telling you that this part is sticking up, and you could really see that it's projecting upwards. And it's, again, you might say, oh, who cares? But it's going to have some significance as we move along in just a few seconds. There's something else I wanted to point out to you, and that is these red structures. These red structures are not typically found in proteins. Proteins are made of chains of amino acids. That's what they're built on. But this particular protein has these additional structures. The fancy name for them is N-acetylglucosamine. You don't have to remember that. They usually just as a shortened way of putting it, N-amino A-G-glucosamine, NAG, which is a fitting word, I think, for a molecule that has negative effects. Okay, but these red regions are believed to make it a little harder for your immune system to recognize these proteins and make antibodies against it. So it's one of the many obstacles that scientists are looking at in terms of working on getting your cells to be able to make antibodies against this protein. Now, some scientists, in some of the literature I've read, have found that there is significant area outside of those red zones to be able to come up with antibodies against this protein. But this is one of the stumbling blocks that scientists have to face. Okay. Okay, so now it's time to look at two other proteins. There is, in the cells that are lining your respiratory system and many other places around your body, you have these two very important proteins. And when I say important, important for what they do naturally, but also important in terms of this disease. One of them is called ACE2. It stands for Angiotensin converting enzyme. This is a normal enzyme which you have in cells in a number of different parts of your body. And as I wrote here, it plays a role in controlling blood pressure. Okay? This turns out to be the major receptor of that spike protein that I just showed you that's on the coronavirus. Okay? So this ACE2 on the surface of many of your cells, including cells lining your respiratory system, those cells have this protein. They also have another protein, which is shown over here. Okay, when I say it, I say TEMPRIS, but it's really a bunch of letters that stand for transmembrane protein serine 2, protease serine 2. I'm not sure why the two S's, but this is an enzyme that also is it's a normal protein that you have in your body. Is involved in certain pathological conditions, certain types of cancers, for example, but it's also involved in some things that are okay, including digestion, blood coagulation, which could be a problem, fertility, inflammatory responses. So these two proteins are natural proteins that you have in your body, and they both play a role in this virus getting into the cells lining your respiratory system. Okay, so let's go a little further with this. All right, so I found this, I need to move your pictures over a little so I could see the screen better. That's not gonna work. Just give me a second. Yeah, I did. Okay, so everybody, This was an article that was in a journal I get called Science, and the authors, which are listed over here, sort of summarize like what happens when this virus attacks you. And I sort of, you could see I cut out, but each bulleted item sort of outlines some of the major steps that the virus takes in infecting you. All right. So it starts with a clear picture is elusive, as this virus acts like no pathogen humanity has ever seen. Okay, so this virus is unbelievable. This is the term that we use to describe the virus. The SARS disease, which happened several years ago, also has a coronavirus. That one they just call SARS-CoV. They don't have a number after it. They didn't have a number because it was the first one, so they didn't need a number. But now, since there's another one, there's actually several other of these viruses, coronaviruses. But the one that's causing problems now is SARS-CoV-2. It enters the nose and throat. The presence of ACE2, which is a protein I mentioned earlier, which normally helps regulate blood pressure, makes tissues vulnerable to this infection. All right. So again, these are normal proteins. The ACE2, normal protein. So it's not like we could just zap this ACE2 and that would make it impossible for this virus to get into our cells. The virus requires that receptor to enter a cell. Once inside, the virus hijacks the cell's machinery, making myriad copies of itself that will then go on and invade other cells, new cells. The virus then marches down the windpipe to attack the lungs. All right, everybody, your lungs have an incredible surface area. You have ACE proteins in other parts of your body. This virus could theoretically attack other parts of your body. But one of the reasons that people think that the lungs is such an issue is that your lungs have a tremendous surface area. I don't know if you could see it, but I took a meter stick, all right? A meter stick is a little over 3 feet long, all right? I'll just sort of hold it. Picture 1 square meter, all right? If I had a surface that was 1 meter by 1 meter, all right, that would be 1 square meter. Your lungs have a surface area of about 70 square meters. It's a field day for this virus, okay? Let me go to the next slide. Let's put these 3 proteins together. We mentioned the spike protein on the surface of the virus, this ACE2 protein, which plays a good role in terms of helping to control your blood pressure, and this protein, I'm just going to call it Tempris, all right? So this protein, when the virus approaches, this protein primes the spike protein such that the spike protein could now attach to the ACE2. Now, I've looked at some literature where they say that at first the spike attaches to the ACE2 and then it gets changed by this protein. So I've seen literature sort of presenting the story 2 different ways. But the bottom line is this is going to allow the spike protein to not only attach, but then to allow this virus to enter into the cell, all right? And it's getting a little late, so I'm going to jump to the next slide. This shows like the overall process a little bit more fully. We don't need to go into all the steps, but let's just look at a few of the important things, at least important in terms of getting a general idea of how this works, all right? Here are the 2 proteins that we were just talking about, Tempris and ACE. Here you see the virus has latched onto the ACE2 protein. It then causes the cell to fold inward, the membrane to fold inward, which is a common thing that cells do. It's sometimes referred to as endocytosis. So it forms this membrane-bound structure around the virus. These are simplified diagrams. I believe that in many cases, when it folds inward, it takes in not just one virus the way they're showing it, but a number of these viruses. The virus then lets out its genetic material, which is going to make all the proteins you need. And there are quite a few proteins that are in this virus. The spike protein is just one of them. It's really beyond the scope of the time we have to discuss all of them. But this spike protein, then the spike protein and all the other proteins come together, which is an amazing thing in itself, to form new viral particles, which are then let out of the cell. And again, it's not just one virus being let out. It's going to be countless viruses let out that will then attack other cells. So that's just a quick summary of what's going on. If we look at this a little further, the thinnest branches of the lungs, I'll just read this with you. You can read it along with me. The thinnest distant branches, of the lung's respiratory tree end in tiny air sacs called alveoli, each lined by a single layer of cells that are also rich in the ACE2 receptors. Normal oxygen crosses the alveoli into the capillaries. Frontline white blood cells release inflammatory molecules called chemokines, which in turn summon more immune cells that target and kill the virus, the virus-infected cells, leaving a stew of fluid and dead cells, pus. When that fluid is filling up the alveoli, you've got a dangerously bad case of pneumonia. This is the underlying pathology of pneumonia with its corresponding symptoms: coughing, fever, rapid, shallow respiration. And it doesn't get better from there. Some COVID-19 patients, that should be a 9, not a 19. Some COVID-19 patients recover. Others deteriorate, often quite suddenly, developing a condition called acute respiratory distress syndrome, ARDS. The oxygen level in their bloods plummet, and they struggle even harder to breathe. On x-rays and computed tomography scans, their lungs are riddled with white opacities where black space, air, should be. Commonly, these patients end up on ventilators. Many die. Autopsies show that alveoli became stuffed with fluid, white blood cells, mucus, and the detritus of destroyed cells. Obviously, I think this doesn't usually happen, but it's happened over 85,000 times in the United States. And I think I said this earlier, but more people have died from this virus in the United States than were killed in the Vietnam War. And pretty soon it may be twice as many. And this is only over a few months. The Vietnam War was over a number of years. So it's scary to say the least. And so I'll just leave you with scientists are working around the world on a treatment to cure someone with COVID-19. And so we're looking for treatments, and we're also looking for vaccines that will prevent people from getting the disease in the first place. I'll just add one little thing. We're almost at 9:20. I will take Q&As if you have, but I'll just add one thing that I made a little bit of a discovery. I'm not a research scientist, but I have taught my kids, I've learned a little bit about computational biology and some of the research tools that are out there. And one of the things that really bothered me, that and to this day it still does, although I know some of the answers now, why some people who get this virus are asymptomatic, 0 symptoms, and at the other end of the extreme, you have people dying from this disease, 0 to death, and everything in between. And that really bothered me. I tried to look for things that might be different in people. And somebody shared an article with me whose child went to SAR a number of years ago. This person works with bacteria that live in your gut. I don't know if you know this, but you have more microorganisms living on or in you than you have of your own cells. And so I thought maybe, that might be tied into it. Maybe, because recently he gave me an article about how these microorganisms have been now associated with Parkinson's disease, which is like a nervous system disease, these organisms living in your gut. And so I won't take you through how I got there, but I eventually started looking at the surface of different bacteria. And I even found one that had a great deal of similarity to the spike protein. And it occurred to me that maybe if some people have more of that bacteria living inside of them, maybe they develop immunity against that virus, which also gives them protection against getting the symptoms of this disease. So I did pass it on to a few scientists. One of them passed it on to some other scientists. So I don't think it's going anywhere, but it is something that I looked into a little bit myself. Okay, I think we're over 9:20. I'll take any questions. If anyone has any questions, I'd be happy to try to answer them. You just have to unmute yourself.
Can you address at what point of the transfer from it entering your body into actually attacking, are they looking to find the cure, either either a vaccine or a treatment? Is it preventing it from attaching onto the ACE2, or like what are they looking at? And what will have success do?
Yeah, there's a lot of different approaches. I'll just tell you a little bit, mainly because I only know a little bit about all the different approaches that are being taken. The ACE2, like some people said, maybe we should block the ACE2, but the ACE2 is like a natural protein in your body that plays a role in controlling your blood pressure. ACE stands for Angiotensin converting enzyme, and angiotensin plays a big role in terms of controlling your blood volume. It's involved in very complex pathways that help you control blood pressure. So you got to be a little careful. Like if you do something to sort of block ACE2 or mess it up in some way, it could have all types of deadly or harmful side effects. From some of the literature I've read, and I don't know if all scientists agree, that other protein that I showed you seems to be one that some scientists feel is a little safer to target in the lungs, that temperance protein. That too is involved in a number of things, around your body. But there seemed to be a feeling that if you were to block something, some natural protein, that would be a better one to block. The best thing, of course, would be to be able to make antibodies that directly attack the virus. And so the spike protein is, a very important one that scientists all over the world are looking for, developing antibodies that will attack that. So that would be, in terms of an immunization. And then, in terms of, in terms of making the antibodies, that would be an immunization. In terms of medication, blocking the temperance protein would also potentially be helpful in terms of treating the disease. That's just some of the many, many, many approaches that different scientists around the world are taking. And I'm sure I only know like a tiny, tiny fraction of the different approaches. Any other questions?
Mr. Goodman?
Goodman, can I just, I'm sorry, go ahead. So another question, go ahead.
Oh, no. I just want to say, Mr. Goodman, nice to see you. Hi, it's Ethan Buren. Thank you.
Oh, hi, Ethan.
Hi.
When Rabbi Hartsock was talking, I see Rabbi Hartsock now, but when he was talking, I was looking at some of the pictures of different people, and I said, wait, those are some of my former students. I'm so glad you were here. I did not see your picture, I will tell you. But it's so nice that you're here.
Hi, Mr. Goodman, I have a question, unless that person had a question also.
Go ahead. Okay,
so my question, it actually relates, I think, to the first picture that you showed. And it indicated that the antigen produces both, stimulates the production of both antibodies as well as memory cells that will produce antibodies. Did I understand correctly?
The picture?
Yes, the memory B cells.
Yeah.
Okay, so the memory B cells will produce antibodies that will attack the antigen also, right?
Yeah,
So
what happens if you got infected a second time, they have receptors on their surface that would recognize that, and they would become plasma cells. There's certain changes they have to go through, but it happens very quickly, and you have a lot of these cells.
So yes, that's what I understood you to say. So I'm not understanding why there's so much concern about reinfection. In other words, for the person who has already had it, the media talks a lot about the antibodies, how long do they actually continue to exist within the body? But we assume those memory cells are still going to be there. So even if the initial antibodies are not there, why are they not relying on the memory cells? I mean, in terms of opening the economy and with the other concerns for those people who have already contracted the disease and recovered from it, why are they not relying on those memory cells in this case?
So I think there's a few things here. One is that that doesn't always happen. For example, like you have to get tetanus shots every so often. They tell you that you should get a tetanus shot like every 10 years. So this, what I showed you with the memory cells, we're hoping, in most cases, we do make memory cells when we're infected, but not in all cases do we make enough to maintain protection for life. And so, I think people are a little afraid at this point since, again, there's a lot of questions about when we should, start, stopping, the social separation and all that. But we can't, we don't know what's going to happen with this virus at this point. The people that were infected were just infected, and they're just recovering now. So whether or not they will retain this immunological memory, we certainly hope it does, that we do, but we don't know for a fact that will be the case. So, there's so many unknowns that are still,
So in other words, it's not only how long the antibodies continue to exist within the body, it's even how long the memory cells exist.
Well, yeah, antibodies are not long-lived. I mean, they're around for a while, but in certain situations, when you have a disease, you're given what's called passive immunity. And there you're actually given the antibodies, and the antibodies destroy the disease, but they don't remain. The bad part about fighting an infection that way is you might not make the memory cells. But for certain diseases that are so toxic, we can't wait to see if you're going to make enough B and cytotoxic T cells to fight the infection. So, if we can give you the antibodies to save you, we're going to do that. All right. But it's different for different pathogens.
I see. And can I ask another question as well?
Sure.
Or does someone else want to turn?
I have no idea.
Okay, well, so I'll proceed. So if I understood you correctly, that the people who unfortunately are dying are actually dying of pneumonia. And
that's mostly true. It's not. There are other things that have been, in fact, some more recent. Like we're seeing, like young children who are getting sick and dying, and in many cases there are other factors. This virus could affect the heart. It could affect other parts of the body as well. So the lung issue is the biggest one for sure, in terms of deaths and really being quite ill from this disease, but it is affecting other parts of the body. This ACE2 protein is in a number of different structures around your body. The virus can't get to some of those places so easily, but it is having effects in other parts of the body as well.
So given that it is often the lungs that are affected, has it become more of a standard protocol to start giving? When a person, I know the disease turns from bad to fatal very quickly sometimes, not always, but sometimes. And so are they giving like antibiotic, like Z-Paks, even though they might not have for a virus, but because they know now that it turns to pneumonia?
As far as I know, there's no drug that is known to work for sure in terms of helping patients. There's some that are being tested. Some of them have, potentially dangerous side effects. I think you heard of this drug that President Trump seems to be taking.
The anti-malarial.
Yeah, and if you go, which by the way has dangerous side effects, and I don't want to get political here, but I can't understand why he's doing it. If you go, there's a site at the National Institute of Health, the NIH, and they have this rather long booklet discussing some of the drugs, discussing the disease in general, but also some of the drugs that are being tested. And so all the drugs have, as far as I know, none of those drugs have been approved by the FDA yet, Food and Drug Administration. And they are being tested in some cases, both animal tests and some of them are at the level of human tests. But they, as far as I know, all the ones I've heard of do have like dangerous side effects, but there are some being tested. But I don't think there are any that are like really known to be that effective. And even some of the ones that you hear about, such as the one that Trump was taking, they say that those drugs should only be taken by people who are already in the hospital, who have like, gotten to the point where they're like very, very ill. And, anything that might help, they're like when doctors are getting desperate, so they're, trying out these drugs when they really need to. Yeah.
Okay, thank you.
I want to thank Mr. Goodman, John Kroll again. I want to thank you for this incredibly enlightening presentation this evening. I learned an incredible amount. I feel like when I read the papers, I'm going to feel like much more knowledgeable in terms of what I'm reading, and I really appreciate what you did for us. So really thank you tonight. That was really great, which I think all of us who are on this call right now kind of wish we were in your AP Bio class, and if this was something of a glimpse of it, we're really glad we could participate. So thank you very much. Thanks everybody for joining.
Thank you.
Thank you so much.
Thank you.
Have a good night.
Good night, everybody.